imagine, this is a very cheap and convenient form of non-invasive brain stimulation. It doesn't require any specialized equipment or training. It's essentially just a magnet placed up to the head. So it's very attractive in that sense for therapeutic use. And it's typically seen in preclinical and preclinical situations to inhibit neuronal activity and reduce cortical excitability. But the mechanism underlying these changes are relatively unknown. And in our lab, we specifically look at axonal plasticity, and we find that changes in axon plasticity can be induced by neuromodulatory interventions. And so we wanted to explore the use of static magnetic stimulation and see, can this actually drive or induce axon plasticity? And when we talk about axon plasticity, we specifically talk about plasticity of the axon initial segment. And this is the part of the neuron that's located at the proximal part of the axon that actually generates the action potentials for neuron communication. There's a very high density of sodium channels that allows neuron depolarization to happen and action potential threshold to occur. And as you can imagine, because this actually generates the electrical signals, it's a very potent regulator of neuron excitability. And what's really interesting is that this axon initial segment is plastic. It can change in response to activity. So just to illustrate this, if we just have that same neuron with the axon initial segment here, when there's periods of reduced neuronal activity, for example, we find that the axon initial segment of individual neurons in response to this will actually lengthen, get longer, or they will actually relocate so that they move closer to the cell body. Now, both of these responses result in increased excitability and makes it more likely for the neuron to fire an action potential. And we also see the opposite effect happen. So when there's periods of increased activity, the axon initial segment of these neurons in response to this can shorten or move further away from the cell body. And this will reduce neuronal excitability. And we think that this is a kind of homeostatic response to activity. So the neuron is constantly fine-tuning its excitability so that it's working within optimal parameters. And then again, as I mentioned, static magnetic stimulation has been seen to reduce neuronal activity. So we wanted to investigate in cortical neurons, does this actually change axon plasticity in some way? And we hypothesized because, again, it seems to inhibit activity, we would expect to see changes that I've outlined here. So either a AIS lengthening or a proximal relocation. So we opted to do this in vitro. So we obtained postnatal day 1 cortical neurons. We seeded these onto cover slips and maintained these in culture for 7 days. And then on the 7th day, we did a media change and the cells were stimulated according to our treatment groups. So one treatment group was just a control where no intervention was applied to the culture well or the media. We had our static magnetic stimulation group, which was delivered by a rare earth neodymium magnet, a 13 millimeter diameter at 0.5 Tesla. And this was placed underneath the culture well, underneath the cover slip, so the magnetic field was in constant contact with the neurons. And then we also had an additional treatment group of 15 millimolar potassium chloride dissolved into the culture media. This is known to depolarize neurons in culture, and it's known to induce AIS plasticity in neurons. And they typically show morphological changes that reduces neuronal excitability. So this is our positive control. And we assess this at different time points. So 6 hours of stimulation to assess the acute effects, 24 hours after this 6-hour period to see if there's any long-lasting or delayed effects. And then also an additional group of 48 hours of constant stimulation to see if there was any dose-dependent effects. And then after this, the cells were fixed, underwent immunostaining, and then we quantified the different parameters of the AIS. So we immunolabeled these neurons by staining for a variety of different markers. So we stained for MAP2, seen in red, to label neurons, Soma and dendrites. For the AIS, we labeled for ankyrin G, which is the main scaffolding protein of the axon initial segment, seen in green. And then we also immunolabeled for inhibitory neurons using GAD65, 67. So all the data I'm going to show you is from inhibitory neurons. And then we used the custom MATLAB script to quantify AIS length. And we manually measured the distance that axon initial segment begins from the Soma. And then we used non-parametric Kruskal-Wallis tests with Dunn's post hoc to see if there was any differences between groups. So now onto the results. So when looking at axon initial segment length, we found that 15 millimolar of potassium chloride stimulation for 6 hours, 24 hours after this period, or for 48 hours of stimulation, significantly reduced the length of the axon initial segment when compared to neurons in the control condition. Again, this has been seen before and is typically seen to reduce neuronal excitability. So it was great that we were able to replicate this. And then for static magnetic stimulation, unfortunately, we didn't see any differences in the axon initial segment length in neurons stimulated with static magnetic stimulation compared to neurons in the control condition. So it seems to suggest it doesn't affect AIS length. And then when looking at the distance from the Soma, we saw that 50 millimolar of potassium chloride stimulation significantly induced a significant distal relocation in cortical neurons with 48 hours of stimulation. Again, this is seen to reduce neuronal excitability, and it has been seen before. And then with our static magnetic stimulation, we didn't see any changes in AIS distance from the Soma with 6 hours of stimulation or 24 hours later. But what was interesting was that we saw significant distal relocation in the AIS with 48 hours of constant stimulation, which was very similar to potassium chloride. So in summary, we wanted to demonstrate, does static magnetic stimulation alter AIS plasticity? And we found some evidence to suggest that it does. And this is kind of the first evidence that has demonstrated this, which is really exciting. Specifically, we saw a kind of dose-dependent effect of static magnetic stimulation. So only when it was applied for 48 hours did we see any changes in axon plasticity. But what was really interesting was that the changes that we saw was very similar to potassium chloride, which is known to depolarize neurons. And the effects that we saw were typically seen to reduce neuronal excitability. So this kind of begs the question, what is this stimulation doing to our neurons in culture? Is it actually increasing activity, similar to potassium chloride? And we don't actually know. So we're following up these experiments by looking at patch clamp, doing patch clamp experiments to see if the morphological changes we see is actually reflective of changes in changes in excitability. And then we're also looking at the mechanisms involved. So we're seeing, is this a sodium or a calcium channel dependent process? So all in all, this is just preliminary data, but it's really exciting and demonstrates that static magnetic stimulation may be able to target axon plasticity in the future. And with that, I'd just like to acknowledge my supervisor, Dr. Alex Tang, and also Miss Emily King, who's presenting after me, and also the various funding bodies and ABSS for allowing me to talk today. So thank you.
Thank you, Jamie. Really, really interesting work. Any quick questions from the group? We've got a minute here for questions. One person asks, thanks, Jamie, how does static magnetic stimulation affect cortical excitability in humans? Do we know that?
So it's typically seen to reduce cortical excitability. Again, it depends on kind of like the parameters, what the actual effect is. It's typically seen to decrease MEPS. It's seen in some cases to induce disruptions in visual searching tasks, but sometimes it's also seen to facilitate some tasks, especially involving the parietal cortex. So again, there's a variety of different effects, and again, the mechanisms are still relatively unknown, but typically it's seen to reduce neuronal excitability or cortical excitability.
Okay, thanks, Jamie. I had a quick question. Given that behavior of the AIS is cell type dependent, you said you looked at cortical neurons. Were these particular types of cortical neurons that you looked at, and do you intend to look at other cortical neurons, interneurons, for instance?
Yeah, so again, like I was saying, we labeled for inhibitory neurons, so all the data was from inhibitory neurons. These were just obtained from just the cortex of postnatal mice. So again, we got have some preliminary data on excitatory neurons as well, so CAM kinase positive neurons, but they don't really survive too well in culture, so we don't have a lot of information on that. But definitely we would like to, again, pursue this more with excitatory neurons as well. We'd like to take this to in vivo as well, just to see if what we're seeing in culture is reminiscent to in vivo. And then again, in our cultures, we do have some glia as well, like astrocytes and things like microglia as well. So we can do a much thorough kind of probe into this to see if those are affected as well.
Great. Thank you. Good work.